for your very excellent presentation as usual. Now I call on for the last, and not the least, of course, speaker, Dr. Yehia Gad. He's a professor emeritus of molecular genetics, and he is the head of the Scientific Committee for Paleobiological Studies in the Ministry of Antiquities. He will be talking about different, not radiology, now the DNA. Wow, it's a very important subject. I'm sure we will be all anxious to listen to his presentation. Please welcome with me Dr. Professor Yehia Gad.
First, I would like to show my real appreciation to the organizing committee of this once-in-a-lifetime event. It's really a great honor to me to share by showing some of highlights about the studies that we have published before and a future perspective of what's coming later on. The field of ancient DNA is relatively much younger than archaeology. It only started about in the mid-80s with the first paper about the quagga, or the horse ancestor, by Higuchi, and the first paper of molecular Egyptology by Svante Pääbo, the Nobel laureate that got Nobel Prize for his pioneering work on hominins a couple of weeks ago. His first paper in '85 was about an Egyptian mummy, actually. In the 90s, the field took off by the PCR, which had the high advantage of amplifying the very low copy number, which is usually in the human remains. It's in a very bad state. So actually, the PCR gave us the privilege and the advantage of ability to analyze this very low copy number and degraded DNA. But actually, it raises another problem, which is potential for contamination with modern DNA, and that's why it necessitated that there has to be ancient DNA authentication guidelines that were adopted by scientific community. And there is about from 9 to 15 guidelines that we need to follow, and we try to follow them all in our work in the Egyptian ancient DNA labs. They have to be a dedicated ancient DNA lab, no modern DNA is allowed in the lab at all. It has to be compartmentalized into smaller spaces, and these spaces should have certain positive displacement air conditioning, UV light to sterilize the air and the walls, antibacterial paint. I mean, there is lots of, it's more sterile than an operating theater in a hospital. So actually, this is, and we usually have protective clothing all the time while we are there. That's why we were in the COVID crisis very well accustomed to using masks. It's not like everybody was really distressed by using masks, but we usually wear masks in the lab for 10 hours or so. Also, there has to be a genotyping of the staff, just to be sure that our DNA does not show in and that's why in the papers that we got, the molecular signature of the lab staff is shown in the paper. And there has to be replication. The same study, probably Professor Zahi referred to this yesterday, the same study has to be replicated. It has to be done by two teams in the two different labs. That's why we had two labs, one in the Egyptian Museum basement, and the other one, which was established in 2007 in the second lab was in Cairo University Medical School, but it has been recently, within the last 3 years, moved into the National Museum of Egyptian civilization. This is the sampling. The sampling has to be sterile, whether it's in the lab or wherever, or inside. This is the sampling of Tutankhamun. And I always use bone marrow biopsy needle to do minimal invasiveness. I can repeat it if you like. Which is 11G. It's like a ball pen tube. It's very thin. It leaves only a very thin hole. And the amount of material that we got is like the pinhead. You see, this is an Eppendorf. This is really tiny thing. So actually, we can generate from this tiny tissue, which with minimal invasiveness to the mummy, a lot of information later on. So the project that between 2008 and 2010 under the leadership of Dr. Hawass was to study the family of the late 18th dynasty, and to try to draw a family pedigree and identify 3 unknown mummies or persons, royals, relying on the knowledge, the previous knowledge of Tutankhamun, the Amenhotep III, and Tuya and Yuya, trying to know who were KV35 elder lady, KV35 younger lady, and KV55. I would suggest Dr. Hawass' next series to make CSI Thebes. It's the same thing. And, but when we started the work, there was that thing that made our life really difficult. Dr. Ikram, Dr. Sahar, Dr. Ashraf, and Dr. Aruli all talked about the resin. And the resin was really pain in the neck to us. If you see here, there is, sorry. If you see, this is the DNA of King Tut. The resin, this is after one of the harshest extraction procedure using what's called phenylchloroform. Still, there is a part of the resin is co-extracted with the DNA in contrast to this DNA, which is for Queen T. Here, this is the way the upper layer should be out, but this was the one for King Tut. And this would make our life difficult because we need to, it kills the reaction. It kills the reaction of the PCR in a very high concentration. I mean, to a great extent. It's 1 molecule per 4,000 for the fetus 1 and the fetus 2, and for King Tut, 1 to 2,000. For 55, it's 1 in 1,000 concentration, and Amenhotep III, 1 in 5. These 4 ladies, they don't have this kind of material. This refers to that the mummification protocols were not uniform. It doesn't, from a chemical point of view, it seems, or a physical-chemical point of view, it seems that the treatments were not the same. So actually, this really is intriguing, and the tight binding of the resin component to the DNA is really intriguing because you know, the DNA is double helix, and if it's tightly binding, somehow it's intercalating with the, presumably, we didn't know this yet, but this need to a chemist or somebody working on mass spectrometry, tandem mass, to see what's the content of this. So we did succeed to separate the DNA using the gel excision assay to cut it out from the gel, away from this inhibitory resin component, and start working with STR, or short tandem repeats, where we use targeted analysis for center genes or loci on the chromosomes. Here we are heading for the Y chromosome, the chromosome for the male lineage, and repeatedly the female gave negative result, and the 7 males that we have studied gave positive results with different degrees of number of loci. The shorter it is, the more loci are, or the more genes are being picked up, but the longer it is, it's being unable to amplify. And we ended up with male lineage, the 3 royal mummies of Tut, 55, and Amenhotep III sharing the same lineage. Yehia is having a different one. This person who were supposed to be, or suspected to be, Thutmose's first, and by the radiology they say it can't be because he has some arrowhead inside, so probably he's not. And also we have another study, Ramses III, for the harem conspiracy, and Unknown Man-E, who proved to be a pentauer. They share the same male lineage, which is different from that of King Tut. So using this male signature, male lineage, using this, which is based on autosomal signature, the minifilar. The minifilar was actually, we had the privilege to use it before anybody else, applied by system, devised this to analyze the very difficult forensic cases. And they have been testing before April 2007 for quite some time with the FBI, and we were the very first people or the lab to try it on a mummy or a human remain, actually. And we could generate data on all studied subjects, as well as we did mitochondrial hypervariable region, which is this one, highly heterogeneous among people, and cloning. And this is the signature of the HVR1 for King Tut in comparison to the reference, the Cambridge reference sequence. It shows 2 different bases here in the DNA, indicating that he has haplogroup K. It ended up that all of the family members share the same mitochondrial lineage or maternal lineage, even Yehia. And I asked Dr. Zahia at that time whether the possibility that Yehia and Tuya are maternal cousins, and he said, I don't think we have historical evidence for that. Amenhotep III is from a different mother, so he has a different mitochondrial lineage, and Hatshepsut and the wet nurse, they don't have the same signature for that. So doing this targeted analysis on the Y chromosome, STRs, the mitochondrial with cloning, and the nuclear or the autosomal loci, we could devise the pedigree into the 4 generation, starting with Yehia and Tuya. We could identify the KV35 elder lady as Queen T, the one who's making all Egyptian women crazy about her hair. How come, my wife was telling me, how come that her hair is that nice? And of course, you all know that Queen T and Amenhotep III were married, and they delivered this guy, most likely his Akhenaton, KV55, and this one too is their daughter and a full sister to KV55. And actually, we didn't so far know, there is no historical evidence who would be the person. I mean, you all know that Akhenaton married Kia and Nefertiti, but who's that? Which one of them, or a third wife? We don't know yet. Also, by targeted analysis, we could detect malarial sequences in 4 subjects, in Tutankhamun, in Tuya and Yehia, and in that a claim to be Thutmose first, but not 2. And this figure outraged some of my colleagues, because some send me emails. Now everybody will say that Tutankhamun died from malaria. We didn't say that. Tuya and Yehia were over 50 years old, I think, right, Ashraf? And they had positive. There is a difference between when you say he died with or he died from. If somebody has a cancer, a terminal cancer, he usually dies with cancer, not from cancer, because usually nobody dies from cancer. Usually the people die from cancer, not from cancer, but from the respiratory infection that happens and kills the person, not because of the cancer, but because of his weakened immunity. So actually, I'm reverberating with Dr. Ruli that we need to be careful about the diagnosis of the cause of death, and that's why we are looking into it. We just mentioned that he had malarial sequences. And this is the old team. Unfortunately, most of them have migrated outside of Egypt after 2011. And now we are moving into a different technology, a very promising technology, a new technology, the next generation sequencing. And mind you, the very first paper that came out using next generation sequencing on an ancient sample was by the Eski Velersif group in 2010. So it's relatively a recent technology. And we are expanding the study of the family by including more persons. This is Amenhotep II, as well as we are revisiting the previously studied. And if you allow me, I will just give a brief statements about the potential of this technology. It's a metagenomic analysis. Actually, it allows to analyze collectively all the genomic material in the sample. So actually, from this very minute amount of tissue, we will be able, inshallah, to get full genomic signature or whole genome sequence of the person. The DNA or the genome of the microbes that have been in the body at the time of death. So actually, this would be only either chronic infection like tuberculosis or malaria or toxoplasmosis, or acute lethal. And that's the way now they are studying, for example, mass graves in Europe. They are studying the plague that happened in Europe between the 14th and the 16th century. So if it's acute lethal and the microbe is still there, we can detect it. Of course, we are not going to be able to detect influenza, because influenza will not be at the time of death in the body to vanish away. So actually, in addition to that, we will be able to detect genomic signature of microbes, saprophytic, that are living on the body, in addition to a new technology that's coming up now, and it's called genomic phenotyping. Genomic phenotyping, it means that to use the genetic variation to draw the picture of a person. Now the FBI is already adopting this, and we wish in the future to use it also to complement the facial reconstruction of using the CT. This is the new team that replaced the old one, and I wish to note this is Professor Sumaya Ismail, my partner in establishing the labs, in training the team, and my colleague and ex-student Rabab Khairat, who had the very first article about using NGS for an ancient mummy in 2013. And these are the younger and my dream team, who are now collaborating with Professor Frank Rulli and University of Zurich in doing our studies. Thank you.
Thanks so much, Professor Yehia. I know that the subject might be a little bit difficult for all of us, but anyhow, this is a very sophisticated technology. Now I'll call on our professors. Please step in for any questions. So please. Now, any questions? I would like to have interactive questions. I'm sure there are a lot. So who will start? Can you please have the lights on, please? People's. Okay. No questions? Okay, I'll start with the first question from my side then. Don't want. There is one here. Okay. Yes, I see you. Can you just raise your voice a little bit, please?
Depression on his skull. Can you explain that?
No, okay. Well, when the